Assalamualaikum warahmatullahi wabarakatuh. Today I will presenting a review on diversity and distribution of mangrove Avicennia spp. The researcher is Binti, Suhaili Binti Cik Malek S50879. My supervisor is Associate Prof. Madya Dr. Abu Hena Mustafa Kamal from Faculty of Fisheries and Food Science UMT. For the introduction, mangroves are considered to be one of the most advanced ecological assemblages of halophytic plants that function as a land-to-ocean transient zone. They consist of taxonomically diverse shrubs and trees spread over tropical and subtropical ecosystems with unique habitats such as beaches, estuaries, tidal creeks, backwaters, lagoons, marshes, mudflats, and even upstream areas where salt water persists. In harsh coastal environments, mangroves are salt-tolerant trees halophytes and are adapted to life. To survive with salt water immersion and wave action, they have a complicated salt filtration mechanism and complex root system. They are suited to waterlogged mud environments that are low in oxygen. In the tropical and subtropical regions of the world, mangroves are available in more than 118 countries and territories. Asia has the world's largest mangrove population, 42% led by Africa, 21%, North America 15%, Oceania 12%, and South America 11%. The distribution of mangrove plants is influenced by different variables. The problem statement, diversity and distribution of mangrove Avicennia spp, studies based on their climatic condition, which is tropical, subtropical, or arid areas are lacking. For objective, to review the diversity and distribution of mangrove Avicennia spp. For the methodology, we highlight 3 points, which is search strategies, study selection, data organization, and reporting. For the search strategy, search articles from databases such as Google Scholar, ResearchGate, Worldwide, Science Direct, and NCBI. Use keywords such as diversity, distribution, Avicennia spp, mangrove, and zonation. All the data was saved at Mendeley desktop and file desktop. For the study selection, filter out all duplicate articles and then accompanied by handwriting to check that non-duplicate articles were present by screening the title, abstract, and taking the full article from the database search. The article was selected. Reports were rejected depends on the inclusion and exclusion criteria. The paper written in English and Malay was approved only. For the data organization and reporting, information was then organized data especially according to the Avicennia species or genus, tropical, subtropical, arid or others, habitat, and authors named that yes. For all the information recorded in analysis study, Prisma guidelines were used. For the result, this table diversity and distribution of mangrove Avicennia spp in different parts in the world. Figure 1 is zonation pattern of mangrove Avicennia spp in tropical region. Figure 2 is zonation pattern of mangrove Avicennia spp in subtropical region. And last figure is zonation pattern of mangrove Avicennia spp in arid region. Avicennia spp is mainly exists in tropical and subtropical region. The diversity and distribution of Avicennia spp including genus Avicennia bicolor, Avicennia germinans, Avicennia marina, Avicennia sauriana, and Avicennia tondesi is highest in America, which is North America and South America, followed up by Australia. Some higher latitude populations, especially Avicennia marina, are also genetically distinct from population in the tropical parts of the range. For the conclusion, studies found that in mangrove swamps and sub-pans, along tidal creeks sheltered by and base and coastline with coral reef ramparts and atoll lagoons, Avicennia is commonly found. Avicennia spp are considered to be pioneer mangrove species in any newly accreted coastal area. This species can tolerate wide range of temperature, hence this species is more dominant in all types of climatic conditions of the world, which is in the form of higher diversity in tropical, subtropical, temperate, arid regions. This mangrove could be an indicator species for climate change research in future. This is my reference. That's all for me. Thank you.